you very much for being here. I want to introduce you to Isabel Fernandes, which is a freshwater ecologist, particularly found of aquatic fungi. Her work focused mostly on understanding how global changes can determine microbial community diversity and activity, and how that impacts freshwater ecosystem functioning. More recently, Isabel is also dedicated to the conservation and monitoring of aquatic fungi through Biodiversity Plus Project, ParAqua, and Most Fun. And she is also the co-chair of the UCN newly founded group on aquatic fungi.
One of them.
One of the two, but the one that I like the most because I know you more. And also Andreas, Bruder, he works on subseed. You did not write it in your presentation. Subseed is the University of Switzerland that is 1 hour from here, and we are very happy for this collaboration. Andreas is a freshwater ecologist with a research interest in functional ecology and how gradients in biodiversity and abiotic conditions affect the functioning of freshwater ecosystems. He applied the knowledge gained from field-based projects for the development of biodiversity monitoring and conservation concepts and initiatives, and you're also the project leader of Most Fun that we are working on. So I give the floor to you. Thank you very much for being here. We are super
Thank you.
Thank you, Lara, for the introduction. And thank you, Lara, for the introduction. Hello, everyone here and online. We will try to make this a little bit brief because try to keep some pace. But if you have any burning question, raise your hand and ask. Otherwise, we can also catch up during coffee or lunch break or so. Andy and I, we will be showing you today a little bit on a different kind of aquatic fungi group. These are not parasites. These are saprophytic fungi. And this is a particular group called aquatic hyphomycetes. And we will tell you a little bit about ecology, story, and also how to culture them, because this is also the main topic for this. And this should be working, but not.
Of the presentation.
Okay, so once upon a time, there was someone that isolated and described what was then the first species of aquatic hyphomycete, and this species was Heliscus lugdunensis, and this was the first species described late in the 19th century by this person here. This species actually was in 2014, if I'm not wrong, renamed after some genetic analysis into a new genus. And this is something that probably will happen a lot with a lot of species because, and I will explain a little bit further down, they are mostly described based on their morphology. And now as we start to have more DNA data, things sometimes change in terms of phylogeny. So this was late 19, and then for some years, things were a bit smooth, cold, let's say. Nothing was happening at a very fast pace. There was another 4 species described, and these are the list of species. You can see here their spores. And just after, another sense, yeah. And it took almost 50 years for the major breakthrough to happen. And this happened with the publication of this paper in 1942 by this person here, which is Cecil Terence Ingold, Professor Cecil Terence Ingold. And he published this paper called Aquatic Hyphomycetes on Decaying Algal Leaves. And as it reads in the introduction, he says, the fungi described in this paper were first encountered as spores suspended in the water in a small stream. And when this water was examined, they found to contain a remarkable collection of fungal spores, blah, blah, blah. And they continue to describe here what can be the typical habitat for these freshwater fungi. So they grow on submerged decaying leaves and usually in well-aerated waters, usually small streams. And in honor of Ingold, these fungi are also known as Ingoldian fungi after his name, freshwater hyphomycetes, and the most typical name, aquatic hyphomycetes. So this is to show you a little bit what we know. This is more or less their cycle, keeping in mind that, too fast. Keeping in mind that this is what we know better, which is their anamorph life cycle. So their asexual cycle, let's say. We know very little so far about their sexual life cycle, their teleomorphs. But for their anamorphs, which is mostly what we study, this is more or less what happens. So they will grow as hyphae. So these are filamentous fungi. They will grow inside the tissues of the plants. And at some stage, they start to develop these structures here. So these are spores, these are conidia, these are asexual spores. And these spores will be released and into the water. And as soon as they touch a new suitable substrate, they will attach, they will germinate, and they keep the cycle. And this is more or less what they look like in real life. So this is a portion of a leaf, and you see all the mycelium growing out a little bit. This is how they look under a microscope. So their ecological role. So when we have this typical ecosystem, so we are talking mostly about very shaded small streams. What happens is that because they are very shaded, the light becomes limiting, so primary production is also limited. So the leaves that fall into these small streams are actually the main source of carbon and energy for a lot of the aquatic biota, including these aquatic fungi. So they are actually the major microbial decomposers of plant litter in streams. Sometimes in other aquatic ecosystems, this can be different, but for these ecosystems, they are the main dominant group here. They have a very extensive array of extracellular enzymes that they can produce that decompose cellulose and pectins and hemicellulose, very, very broad range. And this is one of the reasons why they are so successful. So just a quick overview, here are some data. So they can really have a very large biomass, but also they can be really, really dominant compared to other microbial groups. So there have been some studies measuring productivity, where sometimes they can constitute more than 90% of all the microbial productivity in these decomposing leaf materials. And so because they have this capacity to grow in these leaves, and then there are other higher trophic levels like these shredder species that feed on these colonized leaves, and that increase the quality and the palatability of this resource for these higher trophic levels. They are a very, very important link between basal resources and higher trophic levels in aquatic ecosystems. So they have the capacity not only to acquire nutrients from the leaves itself, but they also have the capacity to acquire nutrients from the water, and they can immobilize these nutrients. So they really are important, of course, because they increase the quality of this material for these invertebrates, but also because they are very important then to mobilize these nutrients. And so they have key roles in carbon and nutrient cycling. So what do we know about their diversity and taxonomy? So we have a bit of a knowledge about these, but we still believe that this is very, very scarce, taking into account that there are not so many experts worldwide working in this group of fungi. And so our knowledge is a bit incomplete. What we know about, in general, aquatic fungi is that currently we have more than 4,000 freshwater fungi species described and more than 2,000 marine fungi described. But the estimates based on molecular data and other estimates is that the total number could be from several thousands to 1 million or eventually even more. So if our estimates are comparable to the ones on terrestrial fungi, we should probably be still below 10% of all the species being described. So there is a lot of work that we can do to help on gaining knowledge on this. And for this specific group, aquatic hyphomycetes, we have nowadays more than 300 species described. But almost on a yearly basis or every couple of years, there are new species being described. So this number will probably keep pacing up and increasing, as we also have the capacity and the experts to work and to look for these species. So this was where I was telling you in the first slide. So this is a little bit how they look like. So they have very different and very beautiful shapes. So these are their conidia. So there are these asexual reproductive structures. They tend to have these tetraradiate shapes or these more sigmoid type shapes. And traditionally, the morphology of the conidia would be used as one of the key features to identify the species. Okay. So the question with the morphology, also together with other components, of course, like a colony color, how they grow, how fast they grow, and so, but really the morphology of the conidia is very, very important. The thing is that if we look to some species, sometimes they tend to look very, very similar. And we are talking about species from different genus. But then when we move to species from same genus, and this is the genus Tetracladium, there sometimes things become really tricky, and it's sometimes really, really hard to have assurance that we are identifying species correctly. And of course, as for many other groups, molecular techniques, DNA barcoding, is really helping us not only to increase our knowledge on the diversity, but also on the phylogeny of this group of fungi. So which region of DNA? This has been mentioned more or less over the last by some of the other colleagues during these days. So there were a few works that started in between 2000, 2010, 12, studying different markers. And this was one of the works that have had a very large number of species tested, and they suggested that ITS could be a good region for these. And then a couple of years after, there was this seminal paper that basically established the ITS region as the primarily barcode for fungi. The thing is that, and then just for an overview, if you have not seen this barcode, so this is within the rDNA cluster. So we have usually part of the 18S, the full ITS1, the 5.8S, the ITS2, and then part of the 28S. So this is what they consider the ITS region. Of course, that this region has some limitation, and it does not work for all the species. So for instance, the genus that I was just showing before, the Tetracladium genus, is one of the examples where the ITS is not good enough to discriminate between the species. And so options that nowadays we can find to cope with this is to go for multi-locus approach. What we have multiple barcode markers to target, to cluster them together and try to get more information, or then to have longer read barcodes where we really sequence a much larger region. And this can be also more informative, not only for identifying, but then eventually also for phylogenetic questions. So a little bit on their biogeography. This was a study that now is almost 10 years. This was a review, a synthesis paper that was done on all the available published papers on this group of fungi so far. So this was between the 40s and the time that they published this paper. And this was based only on information on the morphology of the conidia. So this work did not take into account any DNA or molecular data. And as we can see, there have been species found all over the globe, from the temperate regions, boreal, Arctic, tropical regions. So they are basically spread all over, at least where they have been surveyed. And what the authors found on this paper is that looking at where we find the highest richness, it seems that the highest region is actually on temperate regions. This is a bit contradicting our general perspective that the majority of the richness is on the tropical regions. And then a few years after, there was this other paper, although the number of surveyed locations is much lower on this one. This one was based on DNA, and they also found that the highest regions was more or less complying with the latitude that is, temperate regions. The thing that we would like to call attention here is that, all these areas here, large portions of Australia, large portions of South America, and even here, Canada, they have totally been, never explored. So we should really be paying attention that maybe these trends that we know nowadays from the biogeography of the species, they might have a component of sampling bias. And so it would be really, really important that we make efforts to explore and survey other areas to have a more broad and complete picture about the diversity of this group of species. And why is this important to have an overall perspective of the biodiversity? Because we are living a biodiversity crisis, right? So this is a graphic that was taken from the Living Planet Index. Are you familiar with this index? So this is an index that is produced on long-term data. This is an index that measures populations declines on vertebrates. And this is on vertebrates because we unfortunately do not have this kind of data for other organisms, but we can try to base our knowledge and our trends because probably they will be also the same because the threats that vertebrates are exposed to is probably also the threats that other like fungi and microorganisms are also exposed, like land use change and so on and so forth. And this is the trend that we are seeing. So these are the last data from the report on 24. So since the 70s until now, the average decrease was about 23% on populations of vertebrates. And then when we split these into the different realms, we see that, well, freshwater are very, very, very much affected compared to the other ones. So freshwater species are particularly endangered, and we are losing populations at a higher rate than in other ecosystems. And this could be due to several reasons, and certainly climate change can be one of them. But one of the really big ones is land use changes. So this is what a stream should look like. And this is what you think this is. Yeah, it seems like a highway, but it's not a highway. So this is a river in northeast Switzerland. Yeah, and you can see how much it has changed and how much it does not look as a natural ecosystem would look like. And so all these changes that we humans promote on the ecosystems then have a clear impact on how the species are or the populations are declining in our ecosystems. The other positive side is that, and this is data from Q reports, this is the number of new described species per year. And we can see that we have been increasing this number of species. And this is also true for if we split into the different groups, Ascomycota, Basidia, and early divergent lineages. So this is positive in the sense that we are increasing our knowledge in the diversity, but we need to increase our pace. We need to make these efforts at a faster pace if we want to know who we have in our ecosystem, how many species, where they are, so that we can make efforts to protect them. And that will change.
Thank you.
Welcome.
I don't know if there are questions to this part.
Yeah. Is there any questions? Otherwise, I can make it yet.
Yeah.
We collect them for at the end.
Yeah. So this was an introduction, right? And it's very useful to give you some background because maybe for some of you, this organism group is new. And Isabel tried to explain you why, from different points of view, it's important to understand more about them. Now, given the topic of the school, well, the workshop here, we go a bit into cultures and try to give you some glimpses of what we can use cultures for and how we establish them. This should also be a kind of introduction to what we can do in the afternoon if we have the time. Now then we need to check the program in the afternoon. But it goes into practical topics, right? So around cultures. When we start cultures, well, in terms of what we use them for, it's of course we need them to better describe taxonomy. We can describe taxonomy of certain sites using the morphology of the conidia, like Isabel made some examples. But soon after that, we need cultures to go to establish new DNA sequences. I will show you how many sequences are available for this group of fungi, which is not a lot. But also anamorph-telomorph relationships. So we know them mainly from anamorph life stages. They are involved in decomposition of leaves, as Isabel has said. But another life stage for the same species is the telomorph, and it has different characteristics, different abilities. So to study those, we need to study them in detail. We need those cultures. Then all the other applications that you know from other organism groups, but also ex situ conservation is an important topic for aquatic fungi as well. And Isabel has mentioned quickly the work on the IUCN specialist group that we have started. And at the moment, in terms of cultures, we only have a very small fraction of them deposited. Some are at DSMZ, so DSMZ probably has the biggest collection currently. I don't know if you work with those as well or come across. Well, it's a small anecdote, but probably, sorry?
Yeah. I know we have
bacteria and viruses, but I don't know about fungi have a big collection.
Well, there is a big part, and I think publicly available collections, DSMZ is a good address if you want to get some strains. There is Christian Bachin is the curator for, and she has a strong interest also in research on aquatic hyphomycetes. But of course, if it's not publicly available, then all these applications here are limited. We have our own collection. We share some of our strains, the ones that we sort of finalize and use in publications. We give them, for instance, to DSMZ, replicate. But there is a few research collections somewhere, but they are not fully accessible. So it makes research and exchange a bit more complicated. And with all the so how do we do that? When you want to start a new culture for a species or a set of species, so we go out, we usually pick nice spots. This is in Ticino, but yesterday we were in nice spots just around, well, behind this hill here. So you have nice spots around here too. And unless we have a particular question where we want to have impacted strains, we would go to such a spot because we expect quite a also, it's a forest strain, and so it has a lot of leaves coming in from, as you can imagine, that is forest here. Classically, these are very old techniques. We look for foam accumulations. The foam forms a lot because the leaves release organic substances, and in the foam, the spores are suspended, so we can pick them. They are not germinating, so they are there suspended. They're alive, and the foam doesn't capture many other things, so it's quite a clean sample also, so it helps us to work with. We have some of these samples. If you have time in the afternoon, we can show you how they look like, and
We will make time.
they can talk a bit more about these type of samples. Another type of samples, we can pick the same leaves from the same sites, bring them to the lab, sporulate them like this, with a setup like this, and pick the spores from the water also, well, the conidia. Yeah, and pick them from the individual leaf from the water, plate them, and have monosporic culture starting. If then you don't have contaminations and all these issues, as you know, but it's another technique that also is very efficient. And with these two techniques, we start with an individual conidia, so we know it's an individual that we have in culture. So we look for these conidia. The advantage with this group of fungi is that the conidia are already very specific. So it allows us to identify from a field sample the different species, given the limitations of how closely related morphology are within a genus or between genus. But unlike other microorganisms, we can target some species that we want to have in culture. So we don't randomly isolate, we go targeted. So this is aided by the morphological shape that is very distinct, but also by the size. So we can, well, actually manipulate them. So we would, from these foam samples or from the leaf samples that I've showed on the previous slides, we would pick these individual conidia, place them usually on agar, usually with antibiotics, so we get rid of the bacterial contaminations. And then we observe them and we start seeing them to germinate and to grow, so the start of a colony, well, of a mycelium. And this is the practical steps after isolations, and there we need to have a close eye on it. So that's not something that you can do before you go to holidays. So because there you check if there's contaminations and so on over the next few days. So this will be depending on the temperature, but that's a few days of growth after picking the spore, placing the spore. And also that's the period where contaminations can happen. And that brings me to that topic. So contamination can come from bacteria, can be other filamentous fungi. Often you have yeast also. And especially then long-term storage, if we just put them on petri dishes into the cool room, sometimes we have acaride contaminations as well. So we use antibiotics on these first plates when we isolate and place the conidia, and then later on not, because it reduces a bit the growth of the mycelium. but for bacteria that works. We keep an eye on these isolates and need to transfer or cut out contaminations that we see growing. And for the acarates, there's the potential that we can freeze them, get rid of them this way. Good thing about aquatic hyphomycetes, the culture conditions are very simple compared to probably many of the organism groups that you're working with. So no problem to have them axenic. So this was very simple medium. I will go a bit more detail into the composition of medium that we can use. And there's different types of medium. Some of them it's according to your research question. But overall, it's probably very simple to other organisms. In terms of solid media, that's a few days old and weeks old plates where you can still see those first steps, so the agar plugs that we might have taken from the previous step, which contains antibiotics, for instance. Then we transferred them, agar plugs, colonized onto the new plates, which has better growth conditions and so on. We check for contamination and so on, and then we observe them as they grow. For instance, solid media, we often use multi-extract agar that's very simple. It's containing multi-agar in different concentrations. We use different concentrations for different applications. For instance, if we want to induce sporulation later on, for instance, to confirm morphologically our identification, because we only have seen it once when we picked the conidia in the beginning. We want to confirm. We can do that with the morphology. We put it back into sporulation, and it will produce many spores that we can use for identification. But if we want to just have fast growth of mycelium, for instance, for extracting DNA afterwards or other physiological or biochemical experiments, we can use higher concentration of agar, and then the different sources of nutrients. Solid medium. We can also do liquid medium. That looks like this. So this is pure mycelium growing in a liquid medium. Usually we have either bubbling or shaking to mimic the conditions that they like from the streams in the forest, turbulent and agitated water. We can use them again in experiments, so experiments can run here as well. Different medias used for that, liquid medium. So that's just an example that I took from an experiment, but it's a useful composition. So we use that sometimes when we use a liquid mineral medium. I don't know if that's something that you use with other microorganisms maybe as well. It's quite classic nutrients and vitamins in there, compositions in there, or just a mix which is based on this organic.
MOPS is a buffer, right? MOP, yes.
Mm-hmm.
Yeah.
Okay.
Yeah. And so in terms of if we now think about keeping them permanent, long-term storage, this was sort of the steps that we use for isolation at the start of a culture, but we often want to put them into long-term culture and long-term storage. The principle is that we need to suspend their metabolism, and that's quite logical. We can do that for this group of fungi just by suspending them in water with very low oxygen and nutrient concentration. So we sort of suspended the metabolism like this. I will show you some images of how that could look like. Or, of course, freezing. We often use glycerol for that. So this is suspended in just ionized, sterilized water. It has low nutrient solid medium that we have grown the colony, the cultures on, and we cut a few plugs and transferred them into the water. And that's it. We keep that cool. We have that in a fridge. We have a labeling system and so on, but it's quite simple overall.
Sorry, close. It's just close.
Yeah.
No oxygen exchange.
No. So one of the idea is that you keep low oxygen also because it sort of suspends their metabolism. This is a more simple technique that we have used in the lab. It's based on plastic, so it's probably less ideal than the glass one because maybe sometimes they interact with the plastic of the vial. But we have success with this as well. Another technique that we sometimes try is based on colonized leaves. So we would add during the first steps of a pure culture, we would add some leaf discs, sterilized leaf discs, and let them colonize those leaf discs and then use them in the permanent culture. Because this has advantages that they are still easier to reactivate. They're sort of in the more natural environment compared to the agar plugs. And it has shown based on tests and trials that they are physiologically more natural conditions, so they are more prone to sporulate again when you want to pick them out of the culture collection. So it seems to them that they are doing better physiologically if they are colonized on leaves instead of just on agar plugs. But that's anecdotal evidence and not really in a protocol, I think. We can, of course, freeze them, keep them like this for long-term storage. What can we use the cultures for? So we have them in our long-term culture collection. The applications that we can use them for are very diverse, and you might know them from your own applications. We can make very simple experiments on their physiology under different conditions. So here is an example based on a gradient of temperature. Just we would place them again on solid medium, let them grow under different conditions. Temperature, nutrients can be solid or fixed, but solid or liquid medium, different growth rates under different regimes. This is useful to describe their traits. So we can do temperature growth curves. And it's sort of the start of characterizing the functional potential under different conditions. And that's just a small exploration here towards the start of functional trait databases for these fungi. For instance, their sensitivity to temperature or other environmental gradients, which is interesting. But in terms of aquatic fungi, there's almost nothing in these trait databases.
About 5%. About only 3 to 5% of all the entries on this database is connected to aquatic fungi. Everything is terrestrial. Everything else is terrestrial.
Wow.
And then maybe only for a few traits, right? Maybe there would be many trait categories like temperature sensitivity, nutrient responses, enzymatic potential, but the entries are maybe somebody measured one trait, like a curve like this in an experiment, and put it into a trait data. So a lot of work still to be done there, and it's interesting ecological work, I think. Very relevant also if you think about environmental changes, right? We can use them and use them also for ecological experiments. So this was an experiment that we ran where the question was how different biodiversity levels of different trophic levels. So we have the insects that interact in natural conditions very much with the fungi, and we have the diversity gradient of fungi as well, how these two elements of biodiversity interact, and whether higher biodiversity levels in one or both levels of the food web is beneficial for overall ecosystem functioning. We measure the ecosystem functions here as litter mass, which is classical decomposition rates, which is their main role in the ecosystem, let's say. And we saw that, well, overall, if you have more fungal species, but if you have fungi at all involved, the process rate is, of course, much higher already. If you have a diverse community of fungi, it's higher than the average of a single species. Then if you let them interact with the insects or the more complex ecosystem conditions, then there is a strong positive effect on the process rates. So this is a simple example that I took here, where you can test the interactions between fungal species, but then also in the wider food web context. For instance, we can show that just both trophic levels, so the fungi and the insects acting individually would put the result here, right? The additive response of having both organism groups present. Everything that is above additive is these positive interactions of fungi interacting with invertebrates. In this case, it's the fungi accumulating nutrients, for instance, and having the enzymes to break down a bit of those leaves and making them more palatable for the insects. Another experiment is from Isabel. This is also an experiment where she used one species but two different strains. So she isolated from a polluted stream and a pristine stream and tested their ecological responses to different conditions. Conditions, well, the ecological responses were, again, the litter mass loss, but also the growth of the fungal mycelium under control conditions, and then under impacted conditions, which is a heavy metal in this case. And we see that they respond very differently depending on their origin. So probably there has been some adaptation that they have had to these pollutants. And then if you expose them in a very simple experiment, well, there is other treatment in there, but I take this example here to make it a bit more easy to understand and to follow without wasting too much time. Also, you see these differences, and that's where we see the interspecific difference at the strain level. It's very important and very interesting.
Andy, I just don't want to interrupt, but I think we are running a little bit late. How much time do you think?
Well, let me just make skip a few examples and
Thank you. Sorry, just because
this one is important because it goes back, and I think it's the last one. Let me see. This is important.
Yeah.
And then the afternoon we have time to talk. So.
Yeah. This is important because it goes back to the knowledge of biodiversity and now in terms of barcodes. So of all species described scientifically, and not only with barcodes, these are now globally a bit more than 300 species. So about half of them have a barcode, well, one barcode, ITS. It's the first barcode that we usually characterize. And many of those have only one sequence, so one strain used and deposited the barcode. So nothing is known about interspecific diversity. Nothing is known about the secondary barcode or even the functional genes or the whole genome. So keep that in mind. That's a very small fraction of genetic description for these organism groups. And also it's highly biased globally again, which was the similar bias that Isabel has shown before. Then some examples on ex situ conservation, you will find them in the slides. You can read them up. You also have the publications for this and reintroduction into the environment as a restoration measure. So there's some initiatives. These are examples from terrestrial fungi to put them back into the ecosystem of a disturbed forest here. But the same could be thought of a disturbed stream as a restoration measure to then again reactivate the ecosystem function of this ecosystem. And that's it.